Good morning. Thanks a million for inviting me here today. I'm delighted to present the results on our project that was funded by the EPA. So before I start, I guess I want to introduce our research team. So when I use the royal we, these are the faces that you should be picturing when I'm talking about our research. So it's multidisciplinary. It's from ATU Galway, so Atlantic Technological University Galway, from University College Dublin, and Wageningen University and Research Centre in the Netherlands. Now, the first bit of good news for those of you who had a good night last night, the EPA report is published and all the information that I'm presenting is available online and that you can have a look at that later. Okay, so what were the drivers behind our research? Essentially, when we started out this project, there was very little information on microplastics, particularly in the freshwater ecosystems as opposed to marine ecosystems. So what we decided to do is we'll follow the path. Let's have a look, do some case studies on the sources, have a look at the pathways that those microplastics can potentially take and can they get into our aquatic waterways, and then look at the fate of those microplastics. Are any of them being retained in the freshwater ecosystems, or are they being all washed out to sea? And then we had a list of different recommendations in relation to the different areas that we were covering. So just to give you a quick overview for those of you who may not be familiar with microplastics, essentially what we're looking at is very small particles from 1 micrometre to 5 millimetres. So you're looking at the two orange columns here. So what we would determine large microplastics, so you're looking at the top of your pencil, the rubber that you'd have on the top of your pencil, or even the tip of the pencil. So that's from 1 millimetre to 5 millimetres. But there's also the very small microplastics, and they go down to 1 micron, so smaller than a red blood cell. So there's a huge scale of microplastics that are out there. So it's quite challenging when you're looking at different systems. Also on top of that, we have two different types. We've have our primary microplastics, which people will probably be more familiar with. These are like our microbeads or nurdles, or sometimes known as mermaid tears, and these would have been in relation to the legislation that was brought out for the prohibition of intentionally added microplastics to products. So we'd be familiar with our cosmetics and the Beat the Microbead campaign that was out there. And so we went from actually advertising microbeads in products to now actually advertising that they're free of microplastics. So that was a great first step. But unfortunately, we also have what's known as secondary microplastics. And this is the breakdown of our larger plastic items like our bottles or our textiles. And these will leave behind fibres and fragments and films into the environment. And this is probably the more challenging one to deal with. And this is a lot of what we were finding in our research when we were exploring it. So as you can imagine, the breakdown of plastic items, we're so familiar, it's such, in everybody's life, that there's so many different sources. So what we decided to do is look at maybe one familiar source and two that maybe hadn't been explored before, and do a small kind of case study on that. So the first one we went to was your wastewater treatment plants. So we took three different wastewater treatment plants, and as you can see here on the left, we're in the sunny southeast, and there's no prizes for guessing why we're down in that area if anybody was listening to the talks yesterday morning, but I'll still refer to it as the sunny southeast. So we looked at influent and effluent, and we took samples, and we see, were they doing a good job? And considering that wastewater treatment plants were never designed to capture microplastics, the mean here was up to 82%, and in some samples it was retaining over 90%. And research has shown that they can retain up to 99% in the effluent. Now, we will come back to wastewater treatment plants because any of you out there are involved in farming and biosolids. That's another story that we'll be looking at in our pathways a little later on. Because what was interesting here was that what we were finding as I went through the effluent, the smaller microplastics were the ones that were being released, and the larger, heavier ones were being retained within the sludge and within the biosolids. The next case study we went to was in construction. I think you can see a boom in construction out there, and what we essentially did was we did two things. We went into the lab. Sorry, just take a glass of water. And we created essentially a kill room. So we covered it in a plastic that we could identify. We took components that we know are regularly used in construction. So we used sewer pipe, foul water pipe, and insulation board. And what we did was we used tools that would regularly be used in construction, and we would chop these up into, as you would be resizing for different buildings. And what we found that these were releasing millions of microplastics. And we were looking at the scale of like a small classroom in a school and what was in relation to that. So you can imagine with all the construction that was going on out there. So what we decided to do in conjunction with Carey Construction, we went out to one of their sites and we set little traps, and we were seeing, well, is it being carried away from the construction sites? And yes, it is. So you can imagine if they're like that small, they're going to be carried by wind, they're going to be carried by rain, and they're going to be moved away from the site. Now, the smaller ones travel further, which kind of makes sense, and the larger ones are found near. But we were finding up to, we just went 50 metres from the site. And so then what we did was, well, we did a little infographic. Well, if they're, in the small-scale study that we did, if they are travelling from site, there's lots of different potential pathways that they can take to our rivers and to our aquatic water systems. The other case study we did was on astroturf pitches. So what people mightn't realise is actually like an astroturf pitch is like a plastic woven carpet. And that lovely springy motion when you stand on it is due to the infill, and a lot of the time that's end-of-life tyres that have been ground down, or it's actually designed to contain similar product. It's called SBR. And that's why they use that is that's it's the best material that they can use that can replicate what it would be like to play on a grass turf surface. Okay, and you're looking at player performance, you're looking at safety of people playing within it. So all those little black beads we see here, and I have a little jar of them here if anybody wants to have a look later, are essentially recycled tyres, which we think, fantastic, it's recycling. I could do a whole other story on now how we're actually going to stop doing that in a few years' time, but going forward to see how these then can be transferred. And I think this will be no surprise to anybody who's got like budding Noel McGraths or Dean Rocks or even Messies out there, because if you've got kids or adults themselves playing on pitches, check your car afterwards, you're going to have these lovely little ground microplastics, or they'll come off in your boots or when you're washing your clothes. So now we have got this potential for these microplastics to be, again, put into washing machines, into wastewater treatment plants, have the potential to get into our aquatic rivers. Also the very small ones, because you remember I said they were less than the size of a blood cell, so they can go into the atmosphere as well. So if we have much smaller fibres, even from clothes. So this was showing that, astroturf pitches are a potential source of microplastics to aquatic ecosystems. So now kind of looking onto the pathways. So what we decided to do, well, of these case studies, we look at one. So we looked at agriculture. And thankfully, we've got some great engineers on our project, and they designed this kind of rainfall intensity. So we took one type of soil, we had these packed columns. It allowed different rainfall intensities to go through the column. We used different types of plastics, so different polymers that make up the plastics, and we tried to see could we force those plastics into the sediment. The results were that the plastics remained static. We couldn't get them actually to travel through the column. So what we decided to do then, well, let's see what's happening on land. So off we went out into the field, took some cores, had a look, and yes, we found microplastics that went down to about 35 centimetres. Now that number will be familiar with to some people because that actually coincides with the plough zone depth. So essentially by ploughing in a tillage field, you're actually moving the microplastics from the surface down to a depth of 35 metres. And in the core, you can see here on the left-hand side, the topsoil, that's the soil that's mixed, but below that it's quite packed. So the microplastics aren't, travelling down that deep. So our conclusion was, definitely in tillage fields, that they weren't getting down to the groundwater. They were being retained on the surface. So then we thought, well, what about fields that have a slope? So we found 2 tillage fields that we were allowed to go onto. These both had a record of spreading of biosolids, so back to our wastewater treatment plants. They had a history of spreading biosolids for the last 20 years on their land. So we went to see, okay, now we know that the wastewater treatment plants are obviously retaining a lot of the plastics in their sludge and their biosolids. So now let's see on land that has been spread, is there microplastics and is the slope impacting it? So, excuse me, I think if you close your right eye here, you might have a better view of this graph and you might see a slight decline in field A, but essentially there's no real statistical or no significance in the slope. So again, we were saying, okay, again, this is kind of a snapshot. Let's go back to the lab. Again, we got our engineers, built a bigger system with a rainfall. So these are essentially like 3 clouds here that we can contain the different levels of rainfall that goes in there. There's our table of bare soil, and what we did was then we had different rainfall intensities, so 8 millimetres and 18 millimetres, and we did see that actually the more intense rainfall it was actually carrying the microplastics from the top of the table to the bottom. But what was interesting then is actually, if you seeded that table with grass, so we put in grass swards in there, had two different slopes at 2.5 and 11%, and to see that that have an impact. And what we found was actually the grass swards reduced export potential of the microplastics. So that's quite nice when you think of the acres programme that's out there when they're encouraging grassy verges. So now we know it's actually going to hold on to the microplastics from getting into our aquatic waterways. What we also saw there was slope was shown to be less influential than rainfall intensity in overland microplastic transport. Rivers are a little bit more complex. So we did do a study over 12 months, and you're going to have obviously the levels of the river going up and down. It's quite a complicated process. We did see with high levels the microplastics were diluted. There was no real pattern even with the sediments in relation to the water. So I think more study needs to be done here in relation to looking at a lot more elements within the rivers and looking at a little more intensely. This was a nice element that we had in the project modelling. So this is actually the results of a different EPA-funded project, the Fuse Tools, I nearly forgot, the Fuse Tools project, and they looked at overland flow and looking at the risks if you were spreading, looking at land that was potentially suitable for land spreading of sludge. And so once this was complete, they said, well, what might be nice is actually, could we add another layer to that, looking at microplastics to see, could we actually develop a potential model that if we had enough data that we could input and add that element to see the risk of microplastics getting into the water that are being spread by our biosolids. So here is kind of an example, a hypothetical example of what an output could look like. And again, you can see we're kind of focusing on the sunny southeast down there, and we see a lot of kind of red areas. But this is, as I said, only a hypothetical model. We have to get more data to input within this. But it was a nice kind of build on from the previous EPA project. So kind of moving on to, now I call this environmental fate, and I'll retract it a little bit and call it pathways and environmental fate when I move on. These are our little benthic invertebrates, so all the little creatures that live in the sediment within our rivers. So we looked at different feeding groups. So we looked at ones that filter feed, so ones that would take their food from the water, ones that would gather it, ones that would predate, scavenge and shred. So the names pretty much reflect the different feeding types that are out there. And we found pretty much all of them had up to 73% contained microplastics. So even though they were quite small, they had microplastics, even at a low level. And it's funny, you'll find that number 73 in around 70% for a lot of animals, freshwater, marine. That's the kind of numbers that are coming back between 50 and 80, so kind of a mean of 73%. What was interesting in this, we also took water samples from these sites. So when you look, obviously you can see the big grey bar there in the middle, and that's at site R3. But if you look directly below it, we've got this tall light blue column, and that's our filter feeders, and they're taking plastics from the water. So if we've got more plastics in the water, it's going to show that you're going to have more plastics in the animals that feed from the water column. So it's kind of reflective of what's there. So then if you have ones that are gathering it from the sediment, if there's more in the sediment, they're going to show the higher number. So I thought that was a nice little example to kind of reflect that. We looked at trouts, all of us, and quite generalised, a lot of people eat fish. So it was one thing that we wanted to look at as we were moving up the food chain. So brown trout, again, 72% had microplastics in it. The good thing in Ireland is we remove all the guts and the intestine, and that's where we were recording the microplastics from. So your exposure is very low in relation to brown trout, so don't stop eating trout. As we move up the food chain, we've got a Eurasian otter. And that was kind of the top of the food chain. Now, we didn't go and kill any otters. What we looked at is actually their poo. So we looked at the sprints, and we went around and collected their poo to see could we find any microplastics in it, as you do. And we looked at 3 different areas. Again, we had the sunny southeast as our kind of high exposure area, and we looked at 2 other areas. And again, we found that I think 53% of them had microplastics, but again, what we found is that the most likely pathway is secondary ingestion, so they were getting the microplastics from their food. So we found a lot, we did dietary analysis, and we found they had a lot of salmon, and the salmon too had microplastics in it, and so had the trout. So that's where we think that they're getting their microplastics from. So then we took it to the next level. Now we have information from all the different levels within the food web in the Slaney. So we looked at specific predator-prey interactions. So we did dietary analysis. We looked at the literature, and what resulted from this is that the microplastic uptake stimulated through feeding of benthic invertebrates, and we showed that biomagnification of microplastics is not currently predicted in aquatic biota. So in the majority of animals, and I work in the marine environment as well, they will largely pass it through, so they will retain it for a period. We'll get that kind of snapshot, but they don't build it. So we never see elevated levels of microplastics in the majority of species that we look at now. Mussels, there's a few anomalies within there, but that's essentially that they will pass it through. So you could essentially say, well, it's a kind of a pathway as opposed to an environmental fate. So moving on, kind of, in all these different elements, there is in the report like recommendations in relation to all the different ones in relation to the astroturf, the wastewater treatment plants and all that. But I guess for today, I just picked out one or two, and I looked at kind of recommendations for monitoring. But even that, I think we still need to do a little bit more research before we can say definitively you need to, in a river or catchment, and each one would be individual, where you would sample, where would you monitor, and how would that would reflect it overall. But it would require kind of long-term monitoring of riverine environments. When I say align sampling protocols, now I obviously didn't go into methodologies here, but sediment and water and biota are all sampled, are all processed slightly different. And what you want to do, not so much the actual protocol, but you want to make sure whatever you're using is going to capture a similar level of plastic. So each of the plastics have different polymers of different densities. So you want to make sure that just so you're capturing all the densities, because at the moment there's different methodologies, and even in sediment we mightn't be capturing some of the heavier plastics in there, so we're underestimating what's there. If you're looking at the abiotic matrices, and I guess probably the last one kind of summarises it a little bit better in that your sediment is going to give you a snapshot of what's happening there. Your biota, again, is a snapshot because it's only telling you what is eaten. If something hasn't eaten, you might think this is great, plastics are low, and then you capture someone maybe an hour later and they've eaten and they're going to reflect the microplastic. So it's really only snapshots we're getting from the biota. But the sediment, they are staying there for longer periods. So if you looked at the sediment with the biota that are within the sediment, you get a kind of a nice image. You're taking a bit more of an ecosystem-based approach. And anybody, I guess, who's monitored rivers, you're looking at, are you looking at over a 24-hour period, more intensely sampling to get a better reflection? You're looking at the areas that have high exposure or that are closer to cities or to potential sources of microplastics, and kind of analysing it in that way. So that was just kind of a snapshot of the recommendations that we have. So I just want to say thanks. We have a website where you can access all our different publications, and that there's our contact details, and there's a lot more information on there. If you have any questions, you can just contact us through there. And I have 20 seconds. It's all good.